Chris and I did Project 6 on Bioinformatics Nodosus, and it's where we looked at how the phenotype relates to the genome of this bacterial disease-creating pathogen and the specific differences between the non-infectious and the malignant strains. So first of all, Nodosus causes foot rot in mainly sheep, but also other animals like goats, and affects them in a way that their movements are restricted and can even cause death. Warm climatic environments and moist pastures generally result in the outbreak of this disease. Sheep become more lame as there are signs of inflamed red skin or a grey pasty froth between the digits, which is in this area here. And the skin horn joint is raised, which is between here and here. Problems can also occur depending on the virulence of the infecting strain of Dichelobacter nodosus, in which a strain is a genetic variable of a microorganism, like a virus or a bacterium. These strains can be considered as virulent, intermediate, or benign based on their phenotype or physical appearance. For our project, we use benign, which is this one, and virulent strains in order to find the differences, and we were researching the alterations between proteins. So that would code for a protein, and for both of them, those would be in the ones that the arrows are pointing to, they'd be in the same position and they could code for different ones. And we looked at the function to see if it made an impact in causing foot rot. So an example would be that in this strain, this protein here would code for something that looks at speeding up chemical reactions, but in the virulent strain, that protein codes for a protease which breaks down proteins and peptides. And so these functions are extremely different to one another, and so they would have a substantial influence on how Nodosus has come to be.
Okay, so hi everyone, and I'm going to be talking to you about our method of process. So on the screen here is our method of process flowchart, which is on our presentation, and this was actually made by Famura, so credits go to him for making it. So the first step to our project was the introduction to it. This step was where our group sat together, worked out what we had to do, and how to first look at all the files that Dr. Bullock had given us. We also had Dr. Bullock, the project creator from Monash, briefly explain to us what we had to do. So after our group got a grasp on the task that was at hand, we began our second step, analysing the strains. This is where we began interpreting the strains of the DNA we were given. And this is done by using a program called Artemis, and which is a genome viewer in which we plugged our benign and virulent strains into. The entire strain sequence could then be accessed and viewed within the program as shown in the picture here. And so this is if you scroll along, it's the entire genome sequence of the data we were given. And then after analysing and finding the proteins of interest that we were after, we could plug them into a web-based program called Classical W2, in which we could compare the virulent and benign strains right next to each other to see what the difference is. Next step. So after finding the sequences that we were in our interest on Artemis, we used a program called BLAST. BLAST is a program which acronyms stand for Basic Local Alignment Search Tool, and this program allowed us to compare our unique sequences with a massive database of sequences for a match. So if a match was found, we would be able to view the match and determine from the BLAST results what our actual sequence was. So this was done by entering in our sequences here in the first sequence box and then just hitting the BLAST button. So now we get to go on to obtaining the results. So after hitting the BLAST button, the program would then search for matching sequences that aligned with the sequence we just entered in. After a few moments, a list of matches would appear in the table, shown like this.
So to determine which results align most accurately with our strain, our group checked the tabs of query, cover, and identical over here. So what these tabs here show that the higher percentage in these categories means the more it matches with our sequence. So it's safe to say that this result here would be the most accurate match to our sequence that we used. Yeah. So as we now found a match to our sequence, we then move on to the final stage of researching it. And this could be done by clicking into the result on BLAST or just searching it up on Google. And that concludes our method of process.
So hi guys, I'm doing the summarizing of the results that we found. So we found some interesting differences between the benign and the virulent strains that we were given, particularly among the proteins that the different mutations coded for. Okay, so it's possible, so these are our results. So it's possible that some of the different genes, or rather some of the differences between some of the genes and reference and benign strains, DNO1167 and DNO1034, especially coded for the enzymes that dissolve the hoof in sheep and goats, but more researchers need to confirm this. And with DNO1167, yeah, we suspected that coded for the enzyme that dissolved the hoofs in sheep and goats. Some of the genes that were that were meant to be different were actually the same for both the strains, meaning that the different amino acids still coded for the same proteins. These included various catalysts used for speeding up the reactions inside the bacteria. But unfortunately, most of these proteins were hypothetical proteins, which means these proteins had unknown functions, possibly because there wasn't enough research. So we couldn't go into detail on what these proteins were and if they affected the disease. That's basically a summary of our results.
Okay, so I'm doing the conclusion. And over there we have the end result of the disease, and basically what you see as the disease in the virulent strain. So as you can see there, the hoof has separated from the foot over there, and there's a lot of really nasty stuff happening over there, and it makes the sheep lame, and you can't really do anything with it. And it's also very painful for the animal, which is unethical to limit that way. So to recap, ovine foot rot is caused by Nodosus, as you all should probably know by now. There are two strains, the benign and the virulent strain. The benign strains, actually all of the strains can be killed very easily in dry conditions, or using a foot bath for the animal, and there are a lot of chemicals commercially available that are useful for that kind of purpose. And there is also a vaccine available that gives temporary protection from the disease. So if you are a farmer and a neighbor told you that he has the disease in his sheep, you can use the vaccine and protect your sheep from it. And there are multiple differences in the genome that changes, that is accounted for these proteins that are different. And there are a lot of them with the A suffix hinting at enzymes. Unfortunately, the names were like really long, so it's very hard to understand exactly what they were. And the more information that we tried to find out about it, the less there actually was. There were a lot of websites out there that were basically copies of the original. They don't really provide you with any new information, as we found out, unfortunately. But because of Dr. Bieler, we do know that DNO1167 is the culprit for our virulence factor. It is the one that codes for 3 proteases which are secreted from the bacteria. And these proteases are the ones that account for virulence, basically. Enzyme APRV2 is the specific enzyme that's different between the bacteria that cause the disease. Thank you, sir. It's Nick.